Thank
Awesome.
you. Thanks for helping.
Thanks, Mike.
Yeah, so just push the arrows if you want.
Yep.
Okay.
Good morning, everybody, and thanks for the opportunity to come and talk to you today. And thanks for the technical introduction. You've saved some of my slides. No, you're totally accurate. I've structured my talk today to give an introduction to the technology for those of you who might be new to it, an update on the breadth of applications from a technical point of view, and also some of the updates from London Calling, which was just last week. And I've tried not to put too many of those in that are too technical and complex, because, as Nikki said, it can get a little bit overwhelming at the options that you've got. That's going to be improved over the next 6 months plus as we stabilize the platform and lock in the Kit 14 chemistry and reduce. We're trying to try and reduce the number of choices and options you have, but not the flexibility, because there's been so many ways to do certain things, and it can be a bit overwhelming to begin with. And so we're going to simplify that. So it's going to be getting a little bit better. So why would you use nanopore sequencing? Well, you can access a range of abilities and benefits that you can't get with any other single technology. You can probably get it poorly with a variety thrown in together, but not one single technology will deliver this. Sequencing DNA and RNA directly. I mean, we do sequence amplicons. In fact, we do them beautifully. They've got no modifications on them. You can get huge amounts of data and no length problems, but genomic DNA and RNA is our specialty, including the base modifications. We're short to long read. We're actually read length agnostic. We can go down to 20 base pairs, up to whatever is in your library will be sequenced. Whatever has a motor protein attached to it will be sequenced. And as Nikki said, real-time data analysis, real-time sequencing. So as the pores are sequencing, the base calling is being done, and that data is being generated in a file. And so you can access that very shortly after the sequencing is occurring, and you can make decisions. You can interact with the sequencer. You can stop a run when you've got enough data. If it looks like your library is not as good quality as you were hoping, you can stop the run. You can wash the flow cell and put another library on it. It's very interactive, and that again leads to more flexibility. So this is the core of our technology, and as Nikki said, it's a bacterial pore embedded in the synthetic membrane, and we apply a charge difference across that membrane, and that provides the electromotive force to drag the DNA or RNA through the pore. While that's happening, there's a flow of ions from one side of the membrane to the other, and that flow is disrupted by the shape of the nucleotides going through there. And with really advanced algorithms, we can deconvolute the analog squiggle plot and call bases and the modifications with training. The second component of the system is that purple motor protein. In fact, that's the only necessary library step, is to attach that to the end of your DNA or RNA, and you're good to go. So our library methods are only a couple of hours at most. And that motor protein has three functions. It helps localize the strand at the mouth of the pore. It has helicase activity, utilizes ATP, so it unwinds the double strand. And it also acts as a handbrake to slow the thing down, because without that there, it'll rip through the pore at millions of bases a second, and the best electronics in the world just can't deal with that. So we've got to slow it down to a speed that we can deliver accuracy in base calling. I won't go into any details, but speed through the pore is related to total flow cell output, and we can tweak the type of proteins that we're using and actually increase the speed through the pore. And they're toying with combinations now that can deliver 1,000 base pairs a second and increasing the total output. But we've got to balance that with accuracy, and everything has to increase in its efficiency, not just one thing at a time. But that's for the future. So we assemble multiple pores into three different types of flow cells. The flongle has 126 channels that can measure simultaneously. and delivers a 0.5 to 1 gigabase of output. The MinION flow cell with 512 channels, 20 to 30 gigabases. I think the theoretical maximum's somewhere, but is about 50 gigabases, and groups have gotten over 40, certainly in the field. And the PromethION flow cell is 2,700 channels, so about 5 to 6 times the output from a MinION flow cell. And that generates 100 to 150 gigabases, depending on a few variables in your sample there. Nikki's already put up the family of hardware we've got to run those 3 types of flow cell. The difference between those different boxes is there are 2 differences: the number of flow cells you can run simultaneously and the amount of built-in GPU power. And that's essentially the only difference. So I won't unpack those in any detail, but it means you can choose to use the device that best suits the sort of work you're doing, the size of the flow cell, the number of the flow cells, and whether you need GPU power for base calling or other analysis, or whether you've got plenty of power online in your own IT infrastructure locally. So again, more options, but it means you can find a nice, perfect fit for your scenario. And all of these are available capital free. The pricing there reflects the starter pack reagents. In fact, you can double check online the cost of those reagents because all of our pricing is there. And you purchase the devices by buying a largish annual pack, and the device comes free of charge. In fact, we keep ownership of it. We do have CapEx, and you can buy it if you really, really want to, but there's no real reason in most cases, unless the funding body says you have to own it. And that does pop up occasionally. I really like this slide, and I brought this slide back into use, and it's with some pride. It's now in most of our decks that are used for public facing. It was originally for investors. To explain that Nanopore is a little bit different in the way we do things, and if you don't understand that, it might be a little bit frustrating. And there's 4 areas. So I've already said accessible, capital free. That's a given. Our pricing is transparent. It's on our website. It's the same everywhere in the world. We don't do country discounts, institute discounts, project discounts. That's the price for those products. And that means a little lab in New Zealand or the Broad Institute pays the same price for the same products, which is fair and reasonable, and all companies should do that, but they don't. Upgrades are included. Thank God for that, because we do develop the technology, it advances, and we provide upgrade parts for you at minimum or no cost, depending on what that upgrade actually is. In fact, there's always a constant upgrade of NVIDIA GPU because that technology is evolving, and we constantly want to take advantage of it to provide more rapid, more complex analysis on our systems. Something that's probably known by Nanopore users, but not many other people, is community feedback is part of our developmental process. So we actually release products for use before they're fully developed. Yes, it causes a lot of headaches, but there are some nice advantages with it. So you're not waiting for years for something to be released. And it also means we get feedback on what works well and what doesn't work well. And we make adjustments to the next round of product development based on that. If that variability causes you problems, don't wait till that product's more firm. So we have early release. In fact, we've got developer release if you're really, really, really keen. That's very early on. Then early release, the products have got a shorter warranty period. There's likely to be some improvements and updates, and something might fall over, as the P2 Solos had some issues early on. But you have to understand that when you purchase something that says early release in store, when it's fully released, you can bet it's going to be locked in and performing really nicely around the world. So you just, again, choose which options suit you the best. So here's some platform developments that I'd like to touch base on, although Nikki skimmed the surface with these as well. So our accuracy, I'll start with simplex. So that's just this one strand. And that's what we've focused on mostly, but we're evolving a little bit beyond that. And there are three types of base callers: fast, high accuracy, and super accuracy. And the main difference is the amount of GPU required to run these algorithms. So fast is really good for field work. It allows you to see how much data is being generated. And in some cases, that accuracy, 95, 96%, might be perfectly fine for the work you're doing. But then we've got a high accuracy base caller, which is being tweaked and improved. And it's now generating over Q20. So we call it Q20 plus. And that's a little bit more GPU intensive, but not too bad. Then super accuracy is the best of in class for us, but it does require those algorithms are really complex and they require serious GPU grunt. So there's a cost of using that IT infrastructure. So you've got to consider if you need it or you don't need it. But we're going to focus on how to improve high accuracy, so that's a nice sort of sweet spot in the middle there. And you can see in the graph on the right here, the PromethION flow cell's output is really tied to some of the features of the library. The type of DNA it is. Is it PCR amplified or is it native DNA? What's the length of the library? And we find there's an inverse relationship between the average length of the N50 of the library and the total output from a flow cell. So with ultra long, you might generate between 50 and 100 gig per PromethION flow cell. But if you're looking at 10 KB fragments, you can generate 150 to 200 gigabases sometimes. So it does vary. And the second method for looking at accuracy is consensus calling. And with our new Kit 14 chemistry, bacteria are essentially able to be assembled into a single contig with this chemistry alone and no short reads. And Ryan, we put out a publication just recently with some genomes, bacterial genomes that he was able to sequence either with one or two errors that are most likely modifications, not errors, in the base calling, and complete circularized genomes. And human, we're looking at over Q40 for consensus accuracy, 20 to 30-fold depth of coverage. Variant calling has been improving substantially over the last 12 months, and really happy to report SNPs. When I first joined 4 years ago, I didn't think we were even in the ballpark for SNP accuracy. 20-fold coverage or above, you're looking at 99 to 99.99% accuracy for SNPs. And the great thing is we give you access to nearly the whole genome. With short read sequencing, you get 92% at best. There's researchers that feel it's actually lower than that. And certainly across different organisms it is. But in human, we give you access to the full genome. So you get 99 point something and more of them. So it's actually best in class. And there are publications that validate that. Indels have always been challenging, and they are for all technologies for that matter. I learned that fairly recently. I thought Illumina was the gold standard. Nope, they have problems as well. And in fact, when you look at indels in protein coding regions, we're pretty good. We're well over 95% F1 precision and recall at pretty modest coverage, 30, 40-fold coverage. When you move to the whole genome, that does drop. It's around 80 to 90% F1. Clive's talk last week revealed he's got some strategies in mind that is going to increase that accuracy across the genome. So looking forward to seeing how they roll out. And structural variants, 96%, pretty much from 20-fold coverage and above. And that 96% is pretty much the glass ceiling, because we don't have a truth data set that is more accurate than that to compare it against. So I think you can probably assume that's close to 99% accuracy, precision and recall as well. And so with these new higher accuracies across the genome, applications that were borderline previously are now really doable with high quality data. Whether you're looking at clinical samples for whole genome analysis, or you're looking at cows in a paddock a long way from anything, to help with stock selection, and even there's a nice publication on methylation profiles for age of animals, so you can determine quite interesting characteristics. Duplex is now embedded and is a thing. So duplex sequencing is where the second strand, the complementary strand, follows the primary strand through the pore. And that used to happen 1 to 5% of the time in a library, and we didn't really know much about it then. And it became recognized a year or two ago that if you consensus call across both of those, your accuracy jumps up significantly. And so we've been working on ways, mostly with the chemistries, to how to increase that percentage of a library that duplex calls. And we're now significantly higher than that now. And you can see on the right-hand side here is just I like this here is a representation of a part of an IGB, integrated genome browser, that has our chemistry that we had 12 months ago. Just 12 months ago, 49 kit 10. I think it's about 97, 98% single read accuracy. 410 kit 14, again, simplex, you look at 99%, this drop out of those errors, and duplex. It just is a nice visual of what that increased accuracy does look like. And I won't run through this slide in any detail, but only to say duplex analysis is enabled in our new base caller Dorado. Dorado will replace Guppy in the near future. Dorado is available on GitHub, and it will be integrated into Minnow a little bit later this year. So duplex base calling will become easier and more streamlined as part of an analysis workflow. And this is just a quick snapshot of the benefits of having duplex, primarily for genome assembly. That's where it makes the most difference. The other applications, the specifications are already good enough, but for genome assembly, it does make a difference. And that's where E. coli phased human genomes become a reality in a single experiment. So this is a nice summary slide to help you get your head around what am I going to, so if you're starting an experiment, what am I going to choose to do? Simplex or duplex, fast, high accuracy or super accuracy, and what might I need to make that happen? And both Mike and myself are happy to engage in conversations to help you, and Nikki is a master of this by now, I would assume, helping to decide the best pathway through the forest, because there's quite a few tracks you can go down, and you need a little bit of guidance in helping to select the best route to get to your destination as quickly as possible. Methylation detection is becoming increasingly important from a biological point of view, just as we understand more about it. And because we deliver methylation with the sequencing data, there's no additional work required. And bisulfite sequencing was called the gold standard because nothing else was there to challenge it. I'm sorry, we are now more accurate in in-house benchmarking than bisulfite sequencing. Again, you get more of the genome accessed, and it's simple. You don't have to run 2 sets of sequencing runs and do an enzymatic treatment, etc. So it is, it's deliverable now. As well as methylation, this is from London Calling. We're developing methods for training our algorithms on all context modification calling, as well as other modifications. So 6-methyl-A, for example, is available in Dorado now. So we're moving on to more of the complexity that is in the genome. This is a snapshot of those headline figures that I have been talking about for the last 10 minutes. And these have been steadily improving every presentation I give every 6 months. Data analysis is challenging. There's a lot of data and it's complex. And how do you do it? And what resources do I have available? in-house and what do Nanopore give me? And this is just a nice snapshot of the three general approaches. So Epitome is a cloud-based analysis environment that we've created. It's pre-configured. So we've chosen the tools that are embedded in there. It doesn't require command line. And we're slowly filling out the workflows that are available for ease of use. So in the next slide, I'll put it up here now. So we've got these for infectious disease, biopharm, QC. We've got a nice human variation workflow that covers all of the features you'd be looking for. Structural variants, SNPs, modifications, copy number variants. It works really well and it uses best-in-class tools. And there's a single cell workflow there now. And I'm pleased you put up the 10X because I'm going to finish with some slides at the very end because that's one of the nice new applications coming out. Let me see if I can go back there. We've got Epitome Labs. This is a bioinformatics playground, bioinformatician's playground. It's a Docker environment and you can create your workflows and test them, but you do need command line. And then there's always GitHub, which are research group developed tools that are put up there for evaluation. And sometimes they're awesome and you can do really great things with them. And other times they get developed further. It gives access to a much, the very latest in tools development. Obviously goes up on GitHub ahead of anything else. So some sequencing updates. Is everybody here familiar with adaptive sampling? Nodding, good, a few nodding of heads, but I'm going to mention it because it's confusing if no one's explained it to you before. Adaptive sampling is our method of targeted sequencing that does not use any library or wet lab manipulation of the sample. It uses the sequencer to decide what is sequenced, which is really cool. So you define your targets by their sequence and some flanking region, and you download it into the device, and you tell it to sequence what is on this list. You load your library, just a standard library, nothing done to it. You load it into the flow cell, start the run. What happens is in the first couple of seconds, base calling is generated from each strand in each pore. That's compared back to your reference target list. If it's on there, or with a little bit of wriggle room, if it's on there, nothing happens. That whole strand gets sequenced and that data drops into your data set. However, if it's not there, the hardware is told to reverse the polarity of that one pore, eject the strand from the pore, reverse the polarity and continue sequencing. So you end up with a whole lot of 1/200 base pair off target, genome wide, and then full length for the ones that you want. And you can easily get 10 to 20-fold coverage with those on target. This is ideal for obviously enrichment. So the things that you're looking for, you want at higher depth of coverage. For depletion, so if you're doing a microbiome sample and you don't want the host DNA, say don't sequence this host DNA. And balancing, barcode balancing is going to become more important, and it's on its way for enablement. You can access it. It's early days for this. It's not as simple as you would hope it would be, but it's certainly possible to barcode balance so that each of your targets receives sufficient coverage, which is really important for diagnostic applications where you need to make sure you've got a certain depth of coverage to have statistical relevance. And that's going to be used a lot more in the future. One nice little application is reduced representation by sulfite sequencing, methylation sequencing. So it's similar to RSBS, but it's different. So we've got a bed file of 18,000 regions of the human genome, for example. All the islands, shores, and shells on the human genome run on a MinION flow cell will generate 20-fold coverage, 6.3 million targets, compared to bisulfite, which has about half those number of targets. And if you look on the right-hand side, the sample preparation is really simple, and you can change it every day if you want to change that target file. So you can really do it on the fly And there's some nice applications that can take advantage of that. As well, the PromethION will do adaptive sampling, but because it generates 5 to 6 times the amount of data, you get much higher fold coverage. So here's a panel of 202 cancer genes that achieved 120-fold coverage from a single run. And you still get those background reads, 40-fold coverage, which is, for example, you can look at copy number variation across the human genome with that off-target data, and then have your targeted genes for full length. So there's some really nice combinations, and you can actually control which pores on the flow cell are doing adaptive sampling, and you can turn some of them off. And so half the flow cell is doing adaptive sampling and the other half's not. So there's some other permutations there that, again, more choices, more options to do better science. As I said earlier, we are read length agnostic now. This was enabled in our software last year. And so you can choose a toggle in Minnow and go down to 20 base pairs. And this opens up some really nice applications. So cell-free DNA, there's some nice publications coming out on that now, because you get the standard 170 odd base pair fragment that short read will give you, but then you get multiples of that as well, they're in the blood, they're in the sample that people haven't looked at before, and there's information in there that's really useful. FFPE samples are doable, more challenging because cross-linking does limit the movement of a strand through the pore. Isoform expression, I'm going to finish off with talking about transcription analysis, but with the ultra long reads, you can span complex, when I say complex, they're often repetitive regions without complexity that you can't see easily with short reads because they don't map very well. So centromeres, telomeres, etc. And the ultra long reads really improve. A small amount of that makes assembly so much easier to do. Short fragment mode, as I said, is selectable in the software. If you had one library, we've done this in-house, over 250 million short reads can be generated of 200 base pairs in one PromethION flow cell. So there's a lot of data that can come out of that. Our ultra long read kits are really useful for genome assembly and for looking at telomeres and centromeres and complex structural variations. So finishing off with transcription analysis, and I'm particularly passionate about this, and I appreciate that Nikki put up Affymetrix, because I used to be the Affymetrix product manager before Thermo bought them, and I still like that technology. And Nanopore is really enabling some significant improvements over RNA-seq. Every read is a transcript with Nanopore. There is no assembly required. Throw the Allen key away. You don't have to reconstruct your isoforms. Each read is an isoform. And with our direct RNA kit, you get all of the modifications. It's a bit challenging because there's over 140 of them, and we don't have algorithms developed for knowing what they are. They're actually more often recorded as errors in our sequencing. But they're there, and they're there for a purpose. We just have no idea what they are. So if you're starting a PhD on RNA modifications, you've probably got the next 30 years mapped out in front of you. Hopefully it's well funded, but they're doing something. We just don't know what it is. And there's a whole lot of biology to be understood. We are coming to the end of development of our new RNA, direct RNA analysis kit, RNA 004. It's going to replace RNA 002. I've got a slide in following this, but it's completely enzyme free. So it sequences the native RNA from a sample. So there's no reverse transcription, there's no amplification, there's nothing. It's just raw RNA. And previously you needed quite a lot of it to work well. That's been addressed as well to a large extent. We do in the workflow recommend, it's not required, but recommend a reverse transcription step to provide a second strand for stabilization of the RNA in the library. but it's only the native strand with the motor protein attached that actually gets sequenced. So it's just a feature of the assay itself. This new kit's going to be available 2 or 3 months. I'm hoping it's going to hit the early access in store. The reports from in London Calling is quite a few presentations from groups that got early access or developer access to the kit, and they're reporting results very, very similar to what we get in-house, which is always encouraging. So you've got to go through that transition in-house to research community and then wider use. Accuracy's bumped up significantly from, it was around 92% previously, it's around 96%. And the output, because we've increased the speed of the motor protein, it's actually got a new motor protein, has been increased twofold. So you get substantially more data out from a single run. Yeah, there was something else I was going to mention, but that's fine. And also the amount of starting sample that you need, we've titrated and so you can get quite reasonable amounts of data from as low as 50 nanograms of poly-A RNA to start off with. So this is going to be really useful going forward. We've got that's right. We've got new flow cells. So previously, our older RNA 002 used the DNA sequencing pores. We've moved away from that now, and we have a separate RNA sequencing pore, and so there'll be different flow cells to do this assay with. Whereas previously, it's been the same as DNA, and there's, as I said, different chemistry and software because we're getting better at developing algorithms for modification analysis. Mike might even touch on that on his talk later on. I left it out because it gets rather technical on how we do it, but we're doing it in-house, and we're going to make that available to other research groups because, let's face it, we don't have the resources to develop algorithms for 140 modifications, but there's a lot of researchers around the world. So if we do a few of them, and we'll show you how to do it yourself and take it up and run with it. Last year, we released our updated PCR cDNA sequencing kit. And I'm really encouraged by this because it's a real step forward. So it takes advantage of the developments in our genomic DNA sequencing with a fuel fix adapter and a high capture adapter. And those they mean you can generate over 150 million reads from a PromethION flow cell. That's a huge amount of data. You still get the poly-A tail. So a little tool on GitHub called Tail Finder gives you those lengths. This kit gives you more usable data. Previously, the oligo DT primer would prime on intronic regions in pre-mRNA, and it will give you truncated products. And that could make up 20, even 30% of the total library. And that's just wasted data. This less than 1% with this new assay. It ligates onto the very end of the poly-A tail, not oligo DT priming. There's UMIs in there, and we're going to be releasing analysis software to improve counting applications, and it's barcoding applicable. There's PCR there, but it means you can barcode samples. And this is where I think the take home is really good. A PromethION flow cell, 150 million reads, 10-plex of samples onto a PromethION flow cell will generate between 10 and 15 million reads per sample, all full length. That's pretty deep for a whole transcriptome with all the splice variants in there. And the cost of that will be, I'm ballparking it here, $100 or $200 per sample, which is down where microarrays are at, except microarrays don't discover anything new, whereas this will, and it'll give you the poly-A as well. So this should replace microarrays and RNA-seq. It'll take a while to do it as people get used to it. The analysis becomes more streamlined, but I think for the cost, it's right where we need a technology to be. And a secondary application with this PCS 111 kit is for single cell work. and I'd be really happy to speak with anybody later on about our ability to do single cell analysis. It backends, our assay backends the 10x system. You can take your single cell library out of the freezer that you might have done short read on for gene identification, cell identification with expression patterns. You probably got a little bit of biology from that, maybe not many splice variants, but it gives you an idea of some of the biology through pathway analysis. If you use Nanopore, you will get the full length transcriptome from that sample. We can generate with the cell and sample barcoding on a PromethION flow cell over 18 million mapped reads. So they're the usable ones. You get over 120 million reads, but the mapping, because not all of those transcripts will have the right barcodes on them. There's a whole lot of chemistry there that's suboptimal. Not the sequencing, actually, our sequencing, whatever's there, we will sequence. But you end up with over 80 million barcodes for a library. And this enables you to look at, with our accuracy now, genotyping and SNPs in those. And there's some publications coming out now. All of the isoforms, which is more useful in some diseases and some tissues than others, but it's information you wouldn't have got if you just use short reads. And the isoforms actually enable you to look at subsets of cells for identification, where you wouldn't get that just with the 3' gene calling that you do with short read. There's some really cool things. If that's not cool enough, there are some more cool things coming out over the next year or two. Protein sequencing is advancing nicely in the lab. Sure, it's going to be years away before we have it, but it's still coming. ONT sequencing, where we control the strand and the pore and we shuttle it up and down, and you develop a consensus sequence as you read it each time. That's going to be really useful for needle and haystack applications in disease. Liquid biopsy, we're working with cyclomics, and by the end of the year, we will have a validated assay workflow for looking at targeted mutations in blood samples. Smidgine and Plongle are going to be closer to release because we've got a new ASIC that's the smaller than the size of your pinky fingernail, and that's going to enable these applications to be possible. So I just want to finish with, while other technologies might be equivalent to a screwdriver set, different size screwdrivers, but can only unscrew screws. Or it might be a G-clamp that does that thing really well, but it won't do anything else. We give you the full toolbox. You can look at all of the features of the genome with very few or no restrictions on what you can analyze. And I just think that's going to be the way of the future. Thank you very much. Did I keep the time? Oh, I'm over, sorry. I'd like to ask if anybody's going to Queenstown this year. We're having a Nanopore day on the Thursday. You're invited. There's a registration link on our website. Please come along. We've got 9 or 10 presentations, a little bit longer than this, across the breadth of New Zealand. So it'll be really exciting. So please consider coming along and staying an extra day in Queenstown for that.
And I should have mentioned that after the session, I booked a big table downstairs in OGH. So if you really want to get into some things with Warren, you can have lunch with us.
I've got time early. So I'm free between 12 and 3 o'clock today. So I'm happy to sit down and talk science.
And if there aren't any questions, we'll move to Peter's talk because we're a little bit over. But does anyone have any questions quickly as we load up Peter's talk or Warren or online? Stop share for a minute. You are on the desktop, which is this one. Make sure. Hold on 1 second. I hate Zoom. It kills me every time. This one. You could probably just put it in my hand at this stage. Okay. Yeah, I'll let you. up there. Online, can you see Peter's slide? Hopefully. And then if you just click that, then you should be able to go back and forth that way. I don't know why it comes out differently each time. You can do use slideshow, actually, that might work better. Yeah.
That's better.
Thank
We are seeing presenter mode a minute ago.
Thank you. That's great. So this is Dr. Peterson.
Hello, everyone. So my name is Peter Tsai. I am a bioinformatics research fellow up in the Grafton campus under the Department of Molecular Medicine and Pathology. Today, I'd like to share a bit about how we are using long reads in the cancer space in terms of detecting structural variants. So this is something that we couldn't do very well back in the days with short reads. So I like to start with acknowledgement first. So first I like to thank the individuals who donated the tissues to these studies. And our group are the cancer genomic groups led by Professor Chris Prince and oncologist Dr. Ben Lawrence. So the work that I'm presenting today is a collaborative effort with a number of people, especially with Paula Shield, who pretty much did all the sequencing in this project. And me as a bioinformatician, just sitting there waiting for data. And also Annabelle helped a lot with our adaptive sampling and also Nikki as well. So, as mentioned previously, our group in the last few years has been focusing our effort on pancreatic neuroendocrine tumors. While this is the less aggressive form of pancreatic cancer, it's considered rare. And the 5-year survival of just 37%, it's not a very well-studied cancer type, and the actual links between the genomics and the patient outcome wasn't clear at the time. So we encountered these patients and where we collected 3 different tumors. Upon pathological review with multiple pathologists, there were mixed opinions about exactly what kind of cancer it is. So therefore, we turned to genomics as a way to see if we can identify the tumor type with this in this particular patients. So, however, we've done whole exome sequencing, RNA sequencing, expression microarray, methylation array. We did a lot of genomic stuff on this 3 tumors, and compare with this large cancer genome database called the Cancer Genome Atlas database. We couldn't actually find a match with thousands of tumor types. So we actually don't know what this cancer is. But one thing we couldn't do well with short reads is look for structural variants. And this is what my talk will be mostly about. So the motivation comes from, so this is the copy number of the 3 tumors. So on the top, it's basically just a concatenation of all the chromosomes, and the line indicates the inferred copy number for that particular chromosome. As you can see that the 3 tumors are highly similar in terms of the copy number, but they are extremely highly variable, right? So the only differences that we can see among the 3 tumors is the allele frequency on chromosome 8. So on the bottom row, you have the germline heterozygote SNPs allele frequency plotted against the copy number. So the chromosome A is highlighted with the circle. As you can see that the different tumor has a change in allele frequency. So initially we thought the tumor was spread from liver down to pancreas, down to lymph nodes based on the allele frequency change. But in fact, it doesn't really make sense because when talking to the surgeon, this is very unusual ways of cancer being spread. So we actually don't know which one is the primary at this point. So, but the pattern of this aneuploidy in the tumor is so extreme that it must have been linked with some sort of structural change. However, using show reads, we can't detect any structural variant that we think they were real. So, so we kind of just parked this whole thing on the side for a while. But then Nanopore comes to rescue. So there's no, Warren already did a brilliant introduction to adaptive sampling, which is the technique that we have been using to resolve some of those complex structural variants that we are seeing. So, so computationally targeting a region of interest just sounds amazing. So, however, we now need to decide the regions that we want to target. So, so our target are based on copy number change. So what we did is that we made an in-house web application where we can interrogate the copy number of each chromosome, and we can then manually identify, select the region that we're interested in, and it will then output the genomic coordinate that we can use for adaptive sampling. So, so this is all done in R Shiny 8 for visualization copy number. So, for example, in chromosome 5, we see this change, this drop in the copy number throughout the chromosome. So, therefore, we target the junction where we see the actual change, and same for the chromosome. So, for sequencing, we did all this with Annabelle, and so what we used back then was one flow cell per tumor, and also we also sequencing the blood as the control normal. So we ended up targeting about 14 million bases, and the expected enrichment of those regions is about 18 times coverage to up for 40 times coverage. So so this kind of falls in what the Nanopore was kind of expecting in terms of enrichment. While the state is usually not enough for cancer, for sequencing tumor, but we kind of work with what we get. So the analysis is done using a number of different tools. Unfortunately, there isn't a structural variant somatic caller, so we kind of have to try a different germline structural variant caller and this sort of germline filtering using some custom script that we put together. And of the each breakpoints, we then visualize them in the integrated genome browser. So as an example, chromosome 5, where we saw this drop in the copy number using the Nanopore. So those are just the IGT screenshots. Each gray bar is a read aligned to part of the human genome. So if you see a lot of color dots, that means there's a lot of mismatch. If you see a very consistent mismatch across a read, it means that the part of the reads are being mapped to a different part of the chromosome. This usually indicates there is a structural rearrangement of some sort. So as you can see that in the normal is clean, and in the tumor, we're seeing this structural variant, which we couldn't see back in the day when we're using Illumina, because the junction falls right in between within the exome. So it turns out to be a inversion that we now can call that have resulted in this drop in the copy number. So so this is our target across the one of the tumors. So the yellow bit is what we are targeting. And with adaptive sampling, we can now start linking all this together. So we are now finding the blue box are the inversion, and the red one is the deletion. So all this structural rearrangement in this tumor is something that we couldn't see with show reads, which is actually quite amazing. So we are now trying to decipher what all this means biologically. However, this is not the end of the story, because we then went ahead and did a whole genome sequence of one of the tumor using a few flow cells. Well, that we can detect what we have been seeing using adaptive sampling, we are now seeing a lot more copy neutral breakpoints in this tumor. So we are now actually finding a lot more genes that are being affected. So this is all ongoing. So we are now having to work with oncologists just to decipher what all this means in terms of this very unique tumors. So as I mentioned before, the our group is working on pancreatic nuclear tumor over the last few years. And one of the major outcomes of our work is that we found that in those tumor type, there isn't a lot of somatic mutations. It's not being driven by single nucleotide variant in those or anything like that. It's usually being driven by copy number change. So we found the group of patients who have lost more than half of their genome tend to have a poor prognosis, whereas patients that lost just one or few chromosomes, they tend to have good prognosis. But there's also this group of patients kind of in between the two. In between the two, they also have a variable prognosis. So to take this into more clinical use, the moment we got the adaptive sampling, the first thing that we also try is to see if we can also use those off-target reads for copy number work. So it turns out that we can, and work quite nicely with fairly standardized copy number analysis. So on the top row are the nanopore reads from adaptive sampling, and we are just using the off-target reads. So we can completely reproduce what we were seeing before using a lot of show read sequences, which is another nice utility that we can use. We can do with long reads. Through this Commonwealth Neuroendocrine Tumor Research Collaborative, we got some more funding to extend this work. So to actually try and validate whether the pattern of aneuploidy is associated with the patient prognosis or not. So we have to extend this to a lot more tumors. However, to do this, we need to find the cost-effective ways of looking for copy number. So back in the original study, we relied heavily on whole exome sequencing, cancer panel to detect copy number change. So we had to generate fairly large number of data. So we have also tried this, what we call a shallow whole genome sequencing method, where we only generate actually a lot less than 3.8 million that's in the slides. We can actually get away with less than 1 million reads and still call copy number quite accurately. But to our surprise, we also tried the nanopore reads, where we were able to reproduce the copy number pattern with as few as 185,000 reads. So this allows us to actually start thinking about the clinical utility of this technology in cases where we need to provide a quick diagnosis of patients. We can actually check a few tumors onto nanopore and just get the copy number stuff. But if we have a lot of, we have a good collection of tumor, we can actually go with Illumina, where we can pull multiple samples and also detect the copy number change. So we are not expert of nanopore. So this is our first time trying to use nanopore or even long read sequencing on highly aneuploidy tumors. So it's simple, it's fast, and it's a fairly cool thing that you can just change your target on the fly as well. So you could be, so throughout the adaptive sampling, we actually modify our target between different rounds. So because based on what we did in the first round, we are seeing different things. So we actually changed that. And also they're using the off-target reads, we can also call copy number variations in those tumors. Not to forget that methylation is another big thing that we can get with nanopore. So there's a lot of sort of tissue of origin analysis you can do based on nanopore nanopore methylation patterns, which is something that we are looking to do. But of course, we always end up with more questions than we have answered. What is the better way of identifying targets? Are they more systematic, more statistical ways of looking for a drop in the copy number? So there are now some statistical methods that utilize sort of time series analysis to automatically identify regions that show a big change. And those could be potentially be a more unbiased way of looking for targets. Are they sort of optimal region size? So this is our first time of trying to look for structural variant in junctions where they show a big copy number change. So we actually don't know if we are targeting if our target are too big, too small. So, there's always something to play with. So this is the end of my talk. I'm happy to answer a few questions if time's allowed.
Yeah, thank you for the really nice talk, Peter. Maybe I don't understand what, did I get it correct? You used adaptive sampling because you were working on the human genome.
Yep.
You could use adaptive sampling to target specific chromosome.
Yes, very specific region of the chromosome. Yeah.
Because we are working on sequencing the 6 chromosomes and what structural variation might occur between different individuals. So I wondered if I'm trying to think if the similar approach could work.
I think as long as you have the actual sequence or likely, yeah, because it does a real-time mapping to those regions and then decide to continue sequencing or not. So you really need that regions. Yeah, somewhere close by will do. So our target is quite good. We end up targeting about 40 kb. Yeah.
Yes. It's quite a big way, like genetic distance outside of where you actually wanted to look, that you were targeting down to. So, and proportionally, the amount of human genome that you need to target to is actually quite large.
And Annabelle, I think she needed more data from the people that stick for and
so she needed more chromosome, so she then adapted sample depth for like text. So similar kind of thing, but she had a bit of information about great reference to the best. It's similar,
are there any on the chat too? You can kind of see. I can't even use my own computer anymore. I'm gonna stop sharing. Yep. There we go. You can see if there are anyone else from Zoom. You can type it in the chat or just shout out. Okay. All right. Thanks so much. That's awesome. That was really interesting. Okay, and now I think we have Peter. I cannot talk anymore. Alex and John. So you guys have this one, yeah. So we can do of course I need to go back to Zoom. Slideshow, but first we need to share the screen, sorry. Honestly. You guys, this one, is that yours?
This one,
Yeah. This one. Like that.
Yeah.
Oh, why? I'm just gonna close the other tabs. Yeah, I'm gonna close, there's another one. Close that. Okay, should be good. All right.
Sweet.
John and Alex.
Oh, and I've done something. Cool. Can people in chat see that right?
Yeah,
Online?
yeah.
Yeah.
And you just use the right left arrow.
Cool. So, hey everyone, I'm John and this is Alex. We're both PhD students with Dr. Hilary Shepherd, who is back behind you. And we're going to be talking about how we use nanopore sequencing to validate our gene editing. If you have any questions, please feel free to email us or just come up and chat with us afterwards. And before I get started, I want to thank 3 people in particular: Nikki, Annabelle, who is here. Where is Annabelle? Oh, did she run away? Did she? And Chris, who is a postdoc student, a postdoc worker who actually just left on Friday. So he helped with all the bioinformatics that we use. So if you're unfamiliar with the research that our group does, we are focusing on trying to make a gene therapy for a group of skin diseases called epidermolysis bullosa. And in epidermolysis bullosa, mutations and key functional proteins of the skin means that your skin is fragile and falls off quite easily, leading to quite severe blistering and ulceration phenotypes with quite a high disease morbidity. So our gene therapy pathway, what that looks like is first we isolate skin cells from the patients, and then we use CRISPR-Cas9 to genetically correct the patient-specific mutations. We use those gene-corrected cells to grow sheets of skin, and hopefully the plan is to transplant these gene-corrected sheets of skin back onto problematic areas of the patient and restore the protein and hopefully restore the structural integrity of the skin. And obviously a key part of this whole pathway is analyzing our gene edits at the genomic level. Just a quick background on CRISPR-Cas9, if you're unfamiliar. Cas9 is a targetable nuclease, so it causes a double-stranded DNA break almost anywhere in the genome, defined based on the guide RNA, which is a user-defined sequence. So Cas9 nuclease causes a double-stranded break. We can also use Cas9 nickase to induce a single-stranded break, and if you put 2 of these in close proximity, you can cause a staggered double-stranded break. And in human cells, there's 2 main repair pathways of double-stranded breaks. There's non-homologous end joining, which induces small insertion and deletion events, but there's also homology-directed repair, which is our primary pathway, which is used to incorporate specific edits. And this is what we use to correct the patient mutations. And something key I want to emphasize here is that if we're editing 100,000 cells, that's 200,000 alleles. So we potentially have 200,000 editing outcomes in a bulk DNA. So you'll see why that's important coming up. So why do we use Nanopore in particular? Well, our genomic analysis is based on a standard PCR amplification over our target site, and hopefully we capture all our insertions, deletions, and homology-directed repair events in that amplicon. But this is quite complicated, as you can see in this just IGV plot of some nanopore data. One of our patients, for example, has a heterozygous mutation. So they have a deleted allele and a wild type allele. And this is made even more complicated with our editing designs, where we may target a single allele, or we may use nickase to target both alleles. So for us, we really need individual read resolution to get some clarity on what edits we're actually inducing at these sites. And the reason we use Nanopore, or one of the main reasons we initially got into Nanopore and we use it over Illumina sequencing, which is standard in the field, is because we can do our edits and within the week get our sequencing data back. So it's all in our hands, and it's also much cheaper for our application. So here's an example. There are Sanger sequencing software available that can analyze gene edits. These are great for gene knockouts. This is an example for an edited sample where it's reporting 60% of our alleles have homology-directed repair. So we're like, amazing, this is an awesome result. 60% of the alleles have the edit we want them to have. The other 2 alleles are the wild type and the mutant allele. So there's very little going on. When we nanopore sequence this exact same sample, we noticed actually our edits are a lot more complex than we initially thought. And in terms of our validity of reporting our data, Sanger sequencing said we had 60% editing. Nanopore actually suggested we have 30% editing. So that just emphasizes how important this kind of data is for us to accurately represent our edits. In terms of our bioinformatics, we have a very simple bioinformatic pipeline. We use the flongle. We multiplex sequence amplicons on the flongle. We align the sequences to a reference genome using Minimat2, or the human reference genome. And then we could probably create a software or design an algorithm to sort this out for us, but we just chuck our sequences into something called CRISPR-2, which is actually made for Illumina sequencing. But because the nanopore sequencing accuracy is high enough, CRISPR handles it beautifully. Something else here is that if you're really cost savings focused like us, if we're sequencing amplicons from a different point in the genome, we can just chuck a whole bunch of amplicons in the same barcode, do one barcoding reaction, and just separate the amplicons bioinformatically. So that saves costs even more, because when you're using the flongle platform, the enzymes to do your prep is actually the more expensive part. So just what our raw and processed data looks like. This is a successfully edited sample. And if you look at the raw data, just aligned to the genome, I think it's quite elegant how you can just see everything. In terms of what we're actually looking at, all the highlighted nucleotides are our incorporated edit. We can just chuck this into our Espresso and get a really nice, simple pie chart of all the edits that we have. So that actually comes to the end of my part of the talk. It was really short, but Alex uses Nanopore for a slightly different purpose. So I'll hand it over to him.
Thanks, John. All right, I'm Alex, and I've also been working on the EB project with John, and I've been investigating an exon skipping strategy where the aim is to delete entire exons from the collagen 7 gene that have pathogenic mutations. And the reason this is possible is that this gene has a really long repetitive domain where all of the exons are in frame and short. So several of them, or many of them, we don't really know how many yet, can be deleted at the genomic level without any impact functionally on the protein. And so this is a schematic of how it works. One of the patients that I'm working with has a mutation in exon 31. And we've designed 2 guide RNAs to generate 2 double-stranded breaks in the 2 adjacent introns. This promotes the really efficient NHEJ pathway, which results in the ligation of those 2 introns together and the deletion of the intervening exon with that mutation. And this is predicted to restore normal protein expression. So this is some raw nanopore sequencing data of relatively short amplicons, 650 to 700 base pairs across this exon after CRISPR editing. And you can see the majority of reads, well, most of the reads have a deletion spanning the 2 cut sites. And when we chucked this into CRISPR, we found that about 99% of the alleles had some form of CRISPR editing, and 89% of these events corresponded to deletions spanning the entire exon, so highly efficient. And it is worth noting, as John alluded to, how we used to analyze this with Sanger sequencing and then an online tool called ICE. It was really poor at characterizing these larger deletion events, and Nanopore was fantastic in being able to accurately represent what deletions were going on. We then wanted to find out more about the actual repair outcomes, and we found that the most prominent was this really precise deletion of the desired 125 base pair fragment in between the 2 cut sites. And this confirmed that, as well as being really efficient, it was also relatively targeted. Now, one limitation of short amplicon sequencing like this, when looking at CRISPR editing efficiency, is that any editing events larger than that short amplicon are obviously going to be missed. And it's become relatively evident in the literature, and John actually wrote a review on this, that larger CRISPR editing events do occur, and sometimes at worryingly high frequencies. So we decided to perform some long-range nanopore sequencing across the same region. And of course, Nanopore is really fantastic for being able to identify these larger events. And what we found on the left, this is again some more IGV raw nanopore sequencing data, and you can see this is a 10.5 kb amplicon. You've got the small deleted exon in most of the reads, but you also see these larger deletion events spanning several kilobases. And when we actually quantified these events, we found really interestingly that about 10% of these deletions were too large to be found by short amplicon sequencing. And this is quite significant because in the field, standard practice to assess editing efficiency is to just do short amplicon sequencing. So to be missing 10% of the puzzle is quite substantial and could lead to inflated editing efficiency perceptions, and then also compromise downstream analysis. And then lastly, because the strategy generates 2 intronic double-stranded breaks, we wanted to make sure that we weren't perturbing collagen 7 mRNA splicing by disrupting splicing motifs. And to do this, we extracted mRNA from our CRISPR edited cells. We synthesized cDNA and then nanopore sequenced across exons 27 or 28 to 41. And this, again, was really great using nanopore sequencing because we could get an understanding of what exons were spliced together on the same transcript. And again, this is some raw data and IGV, and this is the transcript data or cDNA data mapped to the human reference genome. And this was confirmed at the mRNA level as well, that the exon was missing in the majority of transcripts. And just eyeballing the rest of the data, it looks pretty good. All of the exons seem to be there. There's no intron read through. So at a first glance, it looked like splicing was occurring as hoped. We then quantified the transcripts we were seeing, and this is what we found. So the wild type healthy cells and the patient cells before editing, we found that the majority of the transcripts were as expected, contained all of the exons. We also did find some lowly expressed alternative transcripts that were lacking a few other exons, so 37 to 39 and then 38. In the patient edited sample, then we found that the most common transcript was this one lacking the exon that we had deleted at the genomic level. And we also found these really low frequency, I guess, aberrant transcripts, including this one along the bottom, where it looks like we've activated a cryptic splice site, and a bit of the intron's actually been read with one of the exons. But overall, this was by far and away the most common, and these aberrant transcripts were only present at quite a low frequency, so we weren't too concerned. So that's all I've got. I just want to say thanks to our funders, thanks to Billy, our supervisor, the rest of our supervisory team and our lab, and then again, a big thanks to Auckland Genomics for kind of getting us set up in the nanopore space and also giving us the opportunity to talk.
So if I need something, so you use Flow
Yep.
for sequencing given genome, like they somehow enrich for your region.
We just sequence amplicons. So we do a 1 kilobase PCR amplification and just see. Yeah.
Okay. Okay, so related question, if you find some like whatever, like desirable edits, how would you select it further?
So for our application, we're just going to use bulk edited cells, right? We hypothesize that we need about 20% editing efficiency, or like 20% of gene correction. So there is no downstream selection process for us. We just want to edit, maximize our editing, and then And were you asking in terms of cell, like the cells, or in terms of the DNA? Ultimately, yeah, there is no selection for us, hopefully.
So you're using this like a quality control, like.
Yeah.
The edits that.
Exactly.
At the cellular level, the goal is to try and get enough cells edited so that we target a sufficient number of stem cells, because then those stem cells, when we grow skin, will survive and regenerate, hopefully, the whole epidermis. If we don't target enough stem cells, then the corrected cells will kind of get outcompeted and won't really lead to long-term skin regeneration.
Now, with the bongles,
Yes.
what's your experience being with them? Is it, you get, what sort of data quantities do you get?
So in the few runs that I've done myself, we're looking at about 500,000 past reads on a 1 kb amplicon. We do everything based on reads, obviously, because that's what we care about. We often get, in terms of the pause, we get the threshold like 50 past pause at the start of each. So that would It'd be amazing if that was better, if we could get 100 pores passed.
Still
but-
early access.
Yes, absolutely. But yeah, it's so easy to load. It does exactly what we want, and $100 for a flow cell. It's pretty good.
Yeah.
Mm.
Another question. I don't think there are any in the chat. We can see. Anyone on Zoom want to ask a question?
It's quiet Zoom folks. Okay, thank you guys so much. That was awesome. And now we have Annie West up next. So good.
Yep.
All right, good job.
Anyway.
All right, Morena koutou. Yeah, I'm Annie. I'm a genomics Aotearoa postdoc working in Kim Hanley's lab. And so now we're changing tack, moving from human chromosomes down to my. And hopefully you'll see by the end of this presentation that Nanopore has worked really, really well for us. Just a bit of background to this project. Wouldn't have been possible without our students that we've worked with over the last year. So we've got Esme, Sophie, and Millie. Esme and Sophie were doing a viral investigation with Nanopore with my COGAD, and then Millie was working with me on some more bacterial-based traits. So this project primarily focuses on samples from the Waiwera estuary, which is located just north of Auckland here. And what we're really interested in doing is profiling the microbial and viral ecology and function across the system. And as an estuary system, you have a lot of changing environments and a lot of potential ecological niches that can be colonized. And so what might be useful here is just to take a look at the bottom of the slide here, where we sampled 9 different sites across the estuary, ranging from freshwater through to marine, and in the middle you had this really nice mixing zone of the fresh and marine water where you get a gradient of salinity happening. And of course that changes throughout the day with the tide coming in and out as well. And part of what I'm really interested in looking at is microbial adaptations to salinity within the system. So typically ways in which microorganisms tolerate salt, which is quite a strong driver of community composition out in the environment, is to either just take in salt and be at one with the surrounding environment, or pump it out and keep the salt out of the cell. So you typically only see the salt in approach employed by some halophilic archaea in this particular order of bacteria. And so it's a little less common. What's more common is that you see the salt out approach where the microorganisms are going to be maintaining their osmotic balance by increasing the proportion of organic solutes within the cell. And so what we typically find associated with these traits is that in the salt in approach, you have quite an acidic proteome. So quite a high proportion of those acidic amino acids making up those proteins. And that's really been studied quite well in really halophilic or like really high salt environments. By comparison with the salt out approach, you do tend to see a little bit of increased acidity in the external proteome, so the proteins that are exposed to the salty environment, but it's much less well studied, and there hasn't really been anything done at this lower end of the salinity scale where we're moving from freshwater to marine. So just a bit of background. The only thing I really want you to take away from this is that we did sample water and sediment sites, but we'll be primarily focusing on the water column sites today. We had 276 metagenomic assembled genomes or MAGs that we recovered from our Illumina metagenomic data. And what's really interesting about this data set is that in our water column samples, particularly the saline water column samples, we see a huge dominance of one actinobacteria called Aquiluna within the Microbacteriaceae family. And Aquiluna resides in the Luna cluster and has a really closely related sister species called Rhodoluna. And what seems to be really interesting is that they're doing this dance between these two freshwater sites here and then the rest of our sort of brackish saline marine community. And so what we're really interested in looking at is how those two sister species are undergoing niche differentiation between those two sites across that salinity divide and how they're coping with those two different environment types. In terms of looking at amino acid encoding across the whole estuary, we found that we did indeed get quite a increase in acidic external proteome with increasing salinity. So on the y-axis, we have the proportion of the amino acid encoded, and on the x-axis, you have increasing salinity across the estuary. And so glutamate and aspartate are our two acidic amino acids that increased in proportion in our external proteomes, and then lysine is our basic amino acid that was decreasing in conjunction with that increase in acidity and salinity. So that was encoded by the genomes across all of our mags, and then we subsequently pulled out that Aquiluna mag and then mapped some transcript data back to that, and we find that really nice pattern again with an increasing acidic external proteome. However, our Aquiluna genome was only 80% complete. So while we're seeing some of these really nice patterns, we can't for sure say everything that we want to about this really interesting bacterium that we find dominating the Wairara estuary community. So we went on a quest to get a better genome for Aquiluna and hopefully also Rhodoluna so that we can see some of that really nice niche differentiation pattern happening. And so this is where Millie, Esme, and Sophie really stepped up, and we went out and got some new samples. So we collected just from 3 representative samples across the estuary this time, from site 1, 5, and 9. And Esme and Sophie actually cultivated about 650 isolates across as many plates. So it was a really big project. So we had a lot of stuff going on. I was really hoping to grab some lunar cluster bacteria. However, what we ended up culturing a lot of was Microbacterium instead of Aquiluna and Rhodoluna. However, Microbacterium is still within that Microbacteriaceae family, so it's still an actinobacteria. And it was actually still really prevalent across the entire estuary. So something unintended, but also interesting that we wanted to explore further. So that's where Annabelle and Nikki stepped in, and we said, hey, can we use Oxford Nanopore Technology to sequence our cultivated isolates? So this essentially happened in about 4 batches. I realized I should have put this in a table, but it was midnight last night when I was doing this slide up. So essentially in batch 1, we had 2 different projects. So these are my first 12 isolates that I ran. And then Sophie and Esme's 55 isolates that they ran as part of their viral project where they were looking at integrated prophage. And so that was done in about October last year. We used the old chemistry. So that was the 9.4.1 flow cell, benign flow cell chemistry. And what Sophie and Esme did was extract DNA from our isolates with the boiling method. And then I subsequently did it with a kit, and you'll see why in a second. Because essentially we found that the boiling method just wasn't giving us good enough DNA that we'd hoped for with working with nanopore benign flow cells. And so of the 55 isolates that Esme and Sophie had submitted for sequencing, they were only able to assemble 50 of them. We had quite a variable range of contigs. So some bacteria were able to be assembled all in one contig. Others took quite a few more. And the average coverage was all right, but the completeness of those genomes was quite variable as well. We also obtained 3 gigabytes of base called read data. So in comparison with the DNA extracted from the kit, I just tended to get a lot better values there. So I was assembling in 1 to 4 contigs. I had a much better average coverage because I also submitted less genomes onto that particular run, and my completeness for all genomes was greater than 98%. So as Nikki was touching on earlier this morning, what you put in is what you get out. So we realized that our DNA quality was really important. So we've been working on that actively in the lab. You'd think it would be really easy with cultivated isolate genomes, but it's actually been proven like really hard to get good quality DNA for nanopore sequencing from them. But it definitely improved when we started using the new technology with the PromethION cells and the new 10 kit chemistry. So we had another couple of rounds of nanopore sequencing. So the third one was where I upped my nanopore sequencing to 26 isolates on a PromethION cell, along with some other stuff from Nikki and Esme. And so there were a few things on that particular flow cell, but I was able to get around 33 gigabytes of base code data compared to 3 previously. And so that was really great. It means that I had some really awesome average coverage values. However, my M50 did tank a little bit. And I think that that was really due to the DNA quality again. So we had quite a few sort of smaller fragments that we included in the sequencing. However, apparently 24 of our genomes were 100% complete. I take that with a grain of salt because we know that we haven't done any manual curation on that yet, but we're getting essentially complete genomes on a single contig, which is really amazing data for us to be able to work with now. The last lot of nanopore data that we got back from Auckland Genomics was Millie's run, where she ran 45 isolates, and we did a size selection. So we removed some of those shorter contigs before putting them on the flow cells because we were hoping to get much better pore activation and occupancy with that. So Millie actually managed to get 1.7 terabytes of data off that. So I think it ended up being 1.4 terabytes of base code data, which was insane. So she has a lot of data to work with, and most of her genomes again assembled in one contig. So really, really awesome data to work with. They all, of course, had more than 100 times coverage because you've got 1.7 terabytes of data. But her completeness actually was a little bit lower than mine. So I'm sort of thinking that maybe even though you've got better data coming out the other end, those shorter fragments were quite essential in the assembly process and just filling in some of those gaps. So it'll be interesting to do a bit more comparison of that later this year. So just a few findings. So from this is, Sophie and Esme's project where they were looking at integrated prophage. They essentially were able to only classify 35 of 50 of their assembled genomes, which just goes to show that there's quite a lot of diversity out in the history that we haven't sampled before. So that's really cool. And 33 of those 35 were actually proteobacteria genomes. So we have quite a good understanding now of the proteobacteria community within the Waiwere estuary. These stars on the tree are the coolest part. That indicates where Esme and Sophie were able to identify integrated prophage within those genomes. So we're actually able to get these really awesome viral contig or genome data and actually use that to be able to map back our metagenomic data a little bit better. So we're able to now have a host link for those prophage, and that means that we can mine our metagenomic data that we previously generated a lot better. So you've got this awesome kind of interplay between some of that old Illumina data that you can then improve your analyses on using your new nanopore generated data. And what Millie and I were doing was really digging into the microbacterium metabolism and also salinity across the estuary. So we were able to recover 3 different species of microbacterium. Oxidans was mostly isolated from the freshwater site. And we can see that we do get some niche differentiation happening between those 3 microbacterium species. So with the oxidans, you've got pectin and polyphenol carbohydrate degradation there, or metabolism, that the other 2 species don't seem to be participating in. And we've also got mercury reduction, which I'm going to be having a look at in the next couple of weeks because I literally made this plot last week. So it's going to be really interesting to dig into a little bit more of this data. By comparison, Microbacterium merotypicum, which as the name suggests is typically isolated from marine environments, tends to have a couple of different substances that it degrades for carbohydrate metabolism. So you've got some amorphous cellulose, some arabinin, and we've also got xyloglucans. And this microbiome algeriense seems to be doing a little bit of everything. So I'm quite keen to see what's happening in that genome. I also made this plot at midnight last night, so sorry for the resolution. But essentially, we're seeing really different patterns in our amino acid content across those isolates. So despite sort of being able to identify the oxidant species from freshwater and Merotypicum, typically in the saline environments, a lot of these charged amino acid proportions are staying relatively constant. So it looks like the picture for this is a little bit more complicated than what we'd seen previously with our metagenomic data. And watch the space because I need to figure out what's going on here. And in summary, Oxford Nanopore Technology has been really great for generating not only bacterial genomes, but also being able to isolate prophage viral genomes as well. And we're going to be doing a lot more in that space. So in that, I'd just like to thank again, Nikki and Annabelle. It's a massive theme here, but you guys have been so helpful in getting us started and providing all the information that we needed around, particularly the upgrade to the new chemistry. But it looks like, even with the old chemistry, we were still getting really great data to work with, and it's just only gotten better over time. So that's really exciting. And yeah, everyone from Hanley Lab, Nessie, and Genomics Aotearoa, and thank you for listening.
Yeah, really nice talk, thanks. And also cool to hear how your research has changed since you've been doing from your PhD research. I have a question. So you mentioned that your colleague was getting like 160 terabytes on the purpose. Is that from a single flow cell?
Yes.
Yes.
Yeah, that was from one of the Promethean flow cells from the new P2 machine. Yeah, Promethean machine. Yeah.
Yeah.
The maximum theoretically is what one.
So I think it can be misinterpreted. So I think for that one, we actually, so there's two types of data that come off the any flow cell, which is the kind of standard FASTQ files, which are ATP geophilic quality, and then there's FAST5 files, which have all that rich information about methylation and the squiggle thing. The FAST5 files are very big. So if you look at the amount of data output, sometimes people can complain that maybe that's the data amount with the FAST5 looks huge. So I think for the Promethean for that run, we got something like 140 gigabases of data. So like it was a great run.
It was fantastic. Yeah.
We were like, we can use this.
It was so good. Yeah.
Delivered like 3 times as much data as they said that we would. So that run in particular, we got a huge amount of data. I would say technically there are different ways of like saying how much data.
Yeah, that was all sort of like, the base called data that was generated from those runs. And I mean, you can already tell that we just had a massive increase in the output of data from that.
So the FASTQ is sort of like the first, what you would think of as like 140 gigs or so.
Yeah.
And then the FAST5, and the additional like people.
Yeah, base calling and stuff. Yeah.
So
And that was all with super accurate base calling as well. So that was all provided. Yeah.
And the other thing is that particular flow cell is about this that we even run.
Oh, really? It was a good run. Yeah.
So they have a different number of pores that pop up as alive, I guess, at the beginning of the sequencing run. And that was just a Promethean had numbers. And did you have to wash it to get that result? No, that was
That was just a single one, yeah.
Not even, so you can look around for 72 hours. And so I think we saw that one after like 48. And it was still, it kind of gives, usually you can start seeing that pores start dying off and your fields start dropping. And that one was still going pretty strong. So you can wash it and then use it for a month. So you could use the last that it was like,
Yeah.
And then they'll find them all alive.
It was still 500. Oh, still active.
I think that's like part of the most like interesting part of the technology for me is being able to watch those pores, be alive or be occupied in real time, because that's all available on the computer screen, and so you can see actually how well your run's doing within 10 to 15 minutes.
Continue working from the place.
Yeah.
You can also monitor from home, so we do that.
Yeah.
Too. You can stop the run from home, and you obviously can't have more RNA from home, but
Yeah.
really effective. Yeah. What program did you use? Did you use GD like that last secretary with the stars?
That was with ITOL. Yeah. So just we ran GTDB on our genomes, and then you get the tree alignment from that.
Yeah. And what we're trying to do with most people's data is we put in real time while we're sequencing, we use the high accuracy base calling. And because the computer can't really keep up in real time, we then move the data to Nessie, do super high accuracy base calling, and then deliver kind of all of that.
So that's the standard to actually use. I'd use the medium option in real time, and then do the high quality.
It just depends on what you need. So it's just that the computer we're using, and you need a pretty good computer to be able to do super high accuracy from a pre-methine flow cell that's generating a lot of data. So, yeah. Okay, any other questions on Zoom either from? Okay, thank you, Annie. Some bacterial work. And then now we have Claire Gebby. Yes. She's going to talk about kind of future work. Thank you. You are here. Oh, don't do that. Just stop. It will happen.
Cool. So hi everyone. I'm a first year PhD student at the Liggins Institute. So I'll be talking to you today about my project is rapid genome sequencing for rare disorders. But I'll be talking to you about the wider project that this fits under, which is the newborn genomics program that's being set up. So firstly, what is rare disease? So we define it as a rare disease that impacts 1 in 2,000 people. So on an individual level, it can be quite rare, but there's over 7,000 different rare diseases and more added each year. So on a collective scale, it's actually quite common. And one difficulty, a challenge for people is to actually just gain a diagnosis in rare disease. So this is some survey data taken in New Zealand last year that showed for over half of respondents, it actually took longer than a year to gain a diagnosis. And for nearly a quarter of people, it actually took over 10 years that they had to wait. So once a diagnosis is gained, only a small % have treatment options available as well. And most rare diseases are genetic in origin, with 50% of them in children. So genetic disorders are overrepresented in neonatal and intensive care, and this can be a really stressful and emotional journey for families, as these infants usually have to undergo multiple invasive procedures to determine the illness and to alleviate suffering. So in an intensive care environment, time really matters. So these decisions need to be made on the scale of minutes and hours, which is vastly different from that time scale you saw on the previous slide of months and years for a diagnosis. So genomic methods have become an increasingly important tool for clinical projects. And so you can see that in 2012 was the first paper that really showed the use of whole genome sequencing for rapid diagnosis in pediatrics. And since then, there's been about 20 to 30 studies that have clearly showed the clinical utility of this. So that's in these three dots here. And these papers have shown about a diagnostic rate of about 30 to 50%, which is much higher than the standard rate of about 10% with standard care. So these papers showed that by doing this, you can decrease the number of days spent in the ICU, decrease infant morbidity, and as well lower the costs of hospitalization. So this paper in the corner here from 2018 is quite a classic example of these groups of paper that showed 30% change in management, 26% change in outcome. So this has really been argued it should become standard of care across the board. And the UK were the first to do this in 2019 as part of an actual public healthcare system. So of note as well is most of these papers are actually using short read genome sequencing data, but there's a group at Stanford University that have been setting world records for the fastest time to diagnosis, and they're using long read nanopore sequencing. So they've had set the current world record, I think, in the Guinness World Records is 5 hours, but that was in 2021. So they've actually reduced that even further recently. And also importantly, they showed variant calling performance comparable to the standard short read sequencing studies. So our primary goal is really just to set up this pipeline to sequence critically ill children in ICU that are undiagnosed and suspected of having a rare disorder. And on top of this, we'll be looking at continuing the research methods into analysing the genomic mechanisms of disease. So one way we'll be doing this, we'll be looking at non-coding regions of the genome and more complex structural variants in infants that don't have any obvious pathogenic variants in protein coding regions. And we'll also be incorporating the full-length RNA transcript sequencing. So that can be really useful. And we're hoping to be able to see the splicing patterns that have produced from any variants that are of clinical concern. So this is what we've got set up so far. So there's quite a few different groups involved in this project. So we've got the University of Auckland and the Liggins Institute here in blue, and Lab Plus in green here, which is a clinical diagnostic lab. So we've got the clinical team and the genetic health service as well in the top corner here, and they'll be working directly with the patient families. So they'll provide the referral and recruitment and also go through the consent process with families. So once that's done, the clinical team will collect blood samples, and there'll actually be 2 sets of samples. So one that's sent through the Liggins Institute and a second set that will be sent through the Lab Plus workflow. They'll also be giving us descriptions of the disease phenotype for data analysis too. So we'll get the blood samples and do DNA and RNA extraction and sequencing. So we'll be doing this on the Promethean P2s. And we'll be separating the parents on each flow cell, and the child will be doing on at least 2 flow cells to gain that extra coverage. And so we'll be looking at a minimum 30-fold genome coverage for those. We'll be doing variant calling and curation. And then all of this data, the DNA sequencing data and patient data as well, will be on a secure research environment. And we've got a dedicated server that's set up just for this project for that. So once we have the variant calling and creation done, that data will be sent to a multidisciplinary team for analysis, and they'll be providing a ranking score of any pathogenic variants or likely pathogenic variants that is found. This will then be sent to the clinical team as a research finding because this workflow on the blue is not accredited yet. So that will be sent to the clinical team and then also sent to Lab Plus, and they'll be performing confirmation of any variants that we find. And that will be as an accredited system that uses their standard operating procedures. And that will result in a clinical report. So that clinical report will then be sent back to the clinical team. And any functional studies that we do as well might take a little bit longer. So that will be a secondary research finding that's sent at a later date. So on top of this as well, we also have a collaboration set up with the Victorian Clinical Genetic Service in Australia. So they'll be looking, we'll be sending them de-identified DNA, and they'll be running that through their pipeline as well. And so they use short read genome sequencing data. So we'll be able to compare our data to their short read sequencing as an extra validation in QA. So why are we using long read? It's basically all of the reasons that Warren mentioned earlier. So I've already touched on the increase in speed and the ability to sequence that full length RNA. And we're also using it for the increased structural variant calling and ability to resolve areas of the genome with low complexity. And we're also looking at the epigenome as well. So there's been a few cases where some quite serious medical conditions have been found in infants based on methylation patterns. So some syndromes where typically there'll be a change in the protein coding gene with a base pair change. You can actually find the same or similar syndromes where the methylation pattern's been disrupted, which leads to decreased gene expression, causing disease. As well as this, in the future, we'll also be looking at using adaptive sampling to increase regions of the genome that differ from parents. So when we expect that maybe around 50% of the time, we might not get an initial diagnosis from the workflow. So in that case, we'll be using more research-driven approach and really looking at incorporating different layers of biological information to investigate the non-coding regions of the genome and how they impact disease. So Justin's group have already developed some algorithms for doing this in more common diseases, and we'll be using them here for this project. And this is starting to be done a bit more in the rare disease space. So there's actually been some guidelines that have been produced for interpreting these different layers of information and in non-coding regions. So we'll be following those. That's set up by the American College of Medical Genetics. Cool. So where are we up to? So we've actually got ethics approval already ticked off. We have a Prometheum P2 on our lab bench at the moment, and we're just waiting for a computer to connect it to, which I've been told is arriving today, hopefully. And we've got another P2 arriving later in about a week, I think. And yeah, so they're the solos as well. And then we can actually get started sequencing. So our first steps will be a bit of validation work in QA, and we've got some genome in a bottle reference samples that we'll be doing for benchmarking as well. So there's quite a few different people involved in this project. So I'd like to thank Justin O'Sullivan, who's my PhD supervisor, as well as the principal investigator on this project. And as I mentioned, yeah, there's lots of different groups involved across the Genetic Health Service, Neonatal Intensive Care, Lab Plus, and the Genetic Health Service. We've got Maori governance as well. So thanks.
Anyone on Zoom have questions?
Pretty quiet. That sounds such like a really exciting
Yeah,
point.
to
Yeah,
finish
it's all
things
coming
started.
together. Yeah, that's great. And so what are the first samples that you think, I mean, after QA, do you have, are there patient samples kind of
Not
ready?
Not yet, yeah, I think we'll do the validation work first, and then but we've got ethics approval to start next month, but I think,
You've got some things
Yeah.
going on, so.
Yeah.
Are you going to look at incorporating any automation in this whole?
That's a good question.
Yeah, so we've got quite a few different set up, and we're also using fabric GM, which I didn't mention, but for very integration. Yeah. But we're going to be doing only 3 samples at once. So for like the lab work, we'll be doing individually because it's parents and child. So we don't need high throughput. But that's something we'll look at.
Great. Okay, so we're coming up to our last talk. So Mike, I've just enabled screen sharing, so you can share your screen for your talk. So we have Mike Yarsky, who's also from Oxford Nanopore Technologies. And that seems to be working, and he's going to give us an update on the newest things, I think, and best practices for ligation prep. And yeah, are you good to go, Mike?
Yeah, I'm just making sure I got presentation mode, and because I can see you over your shoulder, it's perfect.
That's good. All right.
Thanks everyone for having us there today. And look, the most exciting part for me is just like listening to Peter, size update, John Hart, what they're doing with the CRISPR-Cas9 stuff, and just doing the amplicons and seeing what's going on with the gene editing, and and then seeing Anne's West examples of the metagenomic stuff. And I'm looking forward to supporting Claire and Justin and getting them up and running on this rapid diagnostics, because it's just a space that I think just needs some attention. Now, I've got a lot of slides today, but of course, Nikki's done a fantastic job of introducing how Nanopore works, so I'm going to skip all that. And then also Warren covered some of my update talks. So I'll be able to focus hopefully on some library prep tips and tricks, as well as just some applications and how to think about using Nanopore, especially if you're coming from short read sequencing. So I will skip through quite a few slides now. So just know that our mission, though, is to have enable anyone to sequence anything anywhere. And we really are trying to make it easy for that. I'm not going to go through these. I'm just going to skip all through the introduction. Just bear with me. But I will pause here because Nikki did make a point that we are changing rapidly. And I think we're at that top end where we finally hit that diminishing returns of where the platform needs to be. When Warren and I joined back in 2019, we are updating the base callers quite frequently. And I guess I'll just put a note here. So just to educate everyone, when I was selling PacBio or supporting PacBio back in 2012, 14, 15, and we were looking at nanopores that emerged with the map access program in 2014, 15, we saw that, you got this great little device that you could plug into a laptop, but you got a bunch of ends and then just a base, and then a bunch of ends and then a base. And what it was the algorithms just needed time to learn and do some machine learning of actually calling the DNA, native DNA, because there's a lot more modifications on the DNA and RNA that was first thought. And so part of the errors with our algorithm was, one, just improving the logarithms, but the other one was actually recognizing the modifications within the all context. And so that's been worked on and improved. And so you're seeing that we've worked on both Guppy and now Dorado. And then also we've evolved pores. And so we're on the generation of the R10 pore, which improves the holopolymer resolution here. So now we're at a pore, I think, in a state where we've got the base callers. We've also modified the motor protein to have a more consistent speed, which also improves the accuracy. And we now are stabilizing all of our chemistry to the pore 10 and the kit 14. Not everything is there yet, but it will be by the end of the year. And so I'll talk about that a little bit later in the slide. But I just wanted to say that it has been a rapid evolution of nanopore technology, but all of this is invisible to the end user. All the end users, no matter what device they have, they just buy the newer version of the chemistry or the newer version of the flow cells. So all the upgrades come free of charge. And the main thing I want you to take home from this is that when you're reading papers from nanopore publications, think about when they did the work. Because if they said that it's error-prone long read and all this other, jibes or lines about the accuracy, it's because they're using the older chemistry, which was good. Even the consensus accuracy on our pore 9 kit 10 was, Q40, Q50 when you did enough consensus accuracy. There's just been a lot of misinformation out in the field about the single molecule, single read, and people focus on that. But as you saw with the CRISPR-Cas9, they need those single reads. So we give you those single reads. So there's many applications where that works. And now we're at Q20 on a single read. That's just fantastic. Okay, so the updates, we just had London Calling last week. And for those of you that are not familiar with London Calling, it is our big launch of the year. So we do two big launches. We do NC Nanopore Community Meeting in the US in December, and we do London Calling, which is a major event in May. And this is where we talk about our major updates. Warren already touched on some of these. I will talk about the simplex just here, and Warren mentioned this right here, is that basically how you prepare your library will affect the output. And then this is going to lead mostly into the main points that I'm going to make today for my talk. Now I'm going to set a timer to make sure I don't go over time. And so if you share the DNA, say like 10 KB, you can walk away. And this is also similar to Anne's metagenomic samples. A lot of metagenomic extractions somewhere between 5 and 10 KB. You could usually walk away from those flow cells with no interactions. But if you want longer read length, say 30 KB, where you want to phase the genome like a human-sized genome, you can fragment it to 30 KB and then just wash and reload every 24 hours or so. And I'll show you some examples of that to maximize the output and also maximize the flexibility with your data analysis. I'm only going to mention duplex in the sense that when you do duplex, you're doing the complement and template strand. And so when you're doing that, you're reducing the overall coverage of the genome, but you're improving that actually to Q30 plus for single molecule, single reads. or that region that you're reading. So there will be some applications you want to do that, some applications you may not. But just to show you the improvement of our read quality, the bottom frame there of the IGV is showing you the kit 10 pore 9, and then one above it is now the simplex of pore 10 kit 14, and then above that is the duplex. And you can see the error rates are pretty much non-existent now when they are aligned to a reference genome. And this is just showing you a table of the output versus which accuracy using fast, hack, and SUP. Now, there is a major change in the most recent version of Minnow, and that means we've gone to a 5 kilohertz sampling rate, which means our hack accuracy is now 99.0. So it's not much use to even go up to SUP these days if you've got a powerful enough GPU to keep up with hack on your device. There'll be some cases where you will want to do that, especially in cancer and somatic mutations. Now, the main thing, I guess, what also Nikki had alluded to as well as Warren, is modifications detection. We now have in Guppy, we both have the CPG island context, and we also have an all context, because I have had customers in the past where they used the CPG methylation when that was the only model. And unsurprisingly, I should say, that a group of peas, a couple of pea plants, wild types and mutant versions, showed up with a methylation pattern suspiciously looking like human. So we're trying to go away from this and have models that are trained to read it in all contexts. So you can use non-model organisms and do this as well. And the other thing, I guess, that's not on this slide is that if you are a developer and you want to look at modifications or modifications that are not in our pipeline, you can register on our website. There's a beta tool release that you can register to access. So this is proprietary ONT knowledge. So you have to register your access to it, and then there'll be some exchanges with our developers, and then you might or might not get access to this. But if you really are looking at modifications that are outside us, we are willing to work with you. So please register your interest on that website. You can see that announcement in the community under updates. Now, just to touch base on platforms, we have a whole range of platforms, which has already been talked about based on your throughput, and I won't say any more about that. But I will say a new feature that's in the new Minnow is kind of cool. So when you have enough data, you can use this run until. Now, this is not read until, this is run until. So if you know you got a metagenomic sample and you only need 100 gigabases of coverage, you can actually set the run to stop at 100 gigabases of past reads, and then you can use that flow cell. The run will stop when it reaches 100 gigabase, and then you can actually use that flow cell for another sample later in the week or the month, whenever, instead of just burning a whole flow cell for one sample. Adaptive sampling has been covered already by everyone, but I will just like to point out we will be upgrading the Mark 1B to a Mark 1D, which will also be compatible with a iPad at some point. This is still in development. And again, you can register your interest if you want to get a Mark 1D that's going to be compatible with the iPad. Now, this will be kind of cool for the people like Laura Urban, who is working out in the field, doing the rare species on-site sequencing, because now with the new Dorado base caller, it's going to be compatible with the M1 GPUs that are in the M2 GPUs that are in the Apple products. Okay, let's get on to the focus of the talk, and that's sample prep and library. So planning your experiments is a little bit different with Nanopore because you get to control how many pores you can use. Do you use a flongle? Do you use a gridiron? Do you use a Promethean flow cell, or do you use multiple Promethean flow cells? And then also, how long do you run it for? We do have different speeds right now. In the past, we did have the 260 base pair, but that's going to go away. It's only going to be 400 base pairs for the DNA for now, but we are tuning and playing around with the idea of running the motor protein at 800 base pairs per second or even 1,000 base pairs per second. But we need to make sure we maintain that Q20 accuracy, because as we've shown, that Q20 accuracy opens the doors and basically makes our platform the basically best in class for sequencing the whole genome. The direct RNA does run slightly slower, and I'll talk a little bit more about the direct RNA a little bit later. And with the run until, you can actually set how many hours you want the system to run or how many gigabases of output. So again, you have full control over this. You can multiplex the samples. And then the main thing that I'm really going to focus on today is really your fragment length. What does shearing have to do with your input, output, and control of your samples? And we, of course, we have different base calling models, but pretty much we're trying to get everyone to go to high accuracy at 5 kilohertz and then use the SUP when required. So when you're thinking about your library prep, a lot of people with short read sequencing in the past have just worked from the top down. They take the raw sample. They have no control over the library prep because the short read sequencing companies dictate the length of that read. And it's all PCR amplified, which PCR amplification strips away a lot of the modifications, which makes the library perform uniformly. Our libraries do not perform uniformly because a lot of our libraries are native DNA. It's just a fact of nature. So it's just going to our websites, talking to myself and the rest of the technical team and understanding how you can overcome some of these challenges. And there are some challenges for some non-model organisms, especially with avian species, which I'm sure you can talk to Annabelle Whitby. She will happily tell you some of her trials and tribulations sequencing birds. But it's not just extract your DNA, library prep, sequence, data, bioinformatics. With Nanopore, you want to work back to front as well. Because if you can see on the center of this slide here, you can see clearly, let me just actually get the annotation going. Get my pointer. You can see here that the extraction kits directly affect the fragment length. And so we really want you to go to our website, look at our extraction guides and protocols, because different extraction kits will affect your fragment length, and the shorter the fragments you put in, the shorter your reads are going to be. So we really want you to get those long reads. And so you actually want to think, well, what do I want to do with the data? Do I want to phase the methylome? Well, then go for longer reads. Do I just want to do structural variants and methylation? Well, then you can actually shear it down to 10 kb and maximize your output. So there's a balance of what do I want to do for my answer and my biological question. And of course, Nikki alluded to that the quality of DNA is really important for nanopore sequencing. So of course you want to make sure your A280s, 26280s are 1.8 and your 26230s are 2. And there's quite a bit of information on DNA handling and QC metrics. The main thing, if you can do it, is size your DNA sample before you do a library prep. The femtopulse is the best, but not everyone has one of those. You can also do pulse field gel electrophoresis. The thing with adjuvant bioanalyzers, they tend to cut off and lump everything from about 10 kb all the way up to 70 kb. So it's really hard to size a library from Nanopore on a tape station, but it's better than nothing. RNA, we really want at least a RIN score of 7 or above because you're trying to sequence the full length RNA. And so this is really critical to have like a RIN of 8 or 9 when using the direct RNA kits, because you'll only sequence what you put in. And of course, we have the metrics for the RNA, and all this is available on the website under the RNA, under the documents in RNA and the RNA DNA handling. I'd also like to point you to the community as well. We have workflows, best practices workflows for structural variation, for human genome assembly, and metagenomic samples, right from extraction to library prep through to data analysis. Please visit the community. It's a really useful tool. If you don't have access to the community, talk to Nikki and she can sign you up. We don't limit the number of people who have access to it as long as they're associated with the Nanopore device. Now, there's a large number of library preps available, and I'll quickly summarize those. It really just depends on what type of accuracy you want, what type of throughput you want, but you can see we've improved all this. For our DNA, we have a ligation-based, which is about 1 hour. And this goes anywhere from 24-plex or 96-plex or single-plex, depending on how many species and the size of your genomes. This is our highest throughput system and our probably the most widely used and most flexible system. We have a rapid kit, which is a 10-minute, which is fantastic for in-field. Also, if you really have reasonably low input, but I'll show you that in a slide. And we have the ultra long when you're doing things like the telomere to telomere or novel species that you're trying to do de novo assembly. If you have really low input, we do have a PCR where you can do like whole genome amplification protocols as well, very much similar to some of the short reads, but you can actually take advantage of the longer reads, but with low inputs. We do have targeted sequencing where you do full length 16S, or you can do Cas9, CRISPR-Cas9, where you can cut out just a region of interest. And with RNA, we can do direct RNA, which actually reads from the 3' to 5', and it reads the RNA molecule directly. Or you can do the cDNA, and this is done by a PCR kit. We also have a method to do direct cDNA using the ligation kit. We have options for automation. Of course, all of our kits are manual because they're made for low throughput, but we also have high throughput solutions. Well, we've partnered with Hamilton, TCAN, Beckman, et cetera. And all these scripts are available either through our website or through the automation vendor themselves. So I guess to get started, Nikki and Annabelle have been fantastic resources for you. But if you really want to go back and refresh yourself, you can go to the getting started portion of our community. There's also a protocol builder. There's plenty of tutorials and videos on nanopore sequencing. And then there's a protocol selector to help you expose protocol. And then there's plenty of protocol libraries. Now, if you have problems getting through this, because there's some of these seem to be redundant, but it's because we're trying to be flexible with the main library prep. So a lot of these protocols are based on the ligation kit. It's just how you tweak that kit to get the type of library you want. So for DNA sequencing, you basically have the LSK, which is their standard ligation kit, which you basically take your DNA, you high molecular weight DNA. The QIAGEN GentraPure Pure Gene Kit is what we recommend from the human samples to human cell lines from 1 million cell lines. Other kits will be more applicable for non-model organisms. Like I know that Omega Biotech does really well with marine organisms because it's got the Omega Biotech, the mollusk kit, is pretty good for non-model organisms because there's a polysaccharide removement step in there. And so you got to tweak the extraction to match the organism you're working with. There's an optional fragmentation depending on what your aim is, and I'll talk more about fragmentation. And then it's just simply an end repair and then a ligation of the adapters. And to barcode, you basically do end repair, add your native barcodes, and then your sequencing adapters. And we do have 384 barcodes coming in the future. Currently, we have 25.6. The rapid sequencing is just for speed and accuracy. It's not quite the throughput as the ligation. And again, there's a transposase complex with the transposase adapters. You will find a transposase site, nick the DNA, add the transposase adapters, and then you simply just a 5-minute add the sequencing adapters in sequence. And there's a multiplex version of that as well. We do have targeted sequencing as well. So the CRISPR-Cas9, you can region of interest, you can divide your CRISPR-Cas9 guides. It will cut out your region of interest, the end repair, and sequence. And then also, of course, we have adaptive sampling and then targeted 16S barcode. To help you make a decision, we do have a decision tree, and I can get a copy of this decision tree and share this with Nikki and Annabelle to make sure that you guys have availability of this. But you can see for the highest outputs, you can look here for ligation. If you want to do targeted, you can do Cas9 sequencing, and you can also do amplicon based, or you can do adaptive sampling. If you have low inputs, you can do the ligation-based sequencing. I would say one thing, if you have a rare sample, we now have, you can load a flow cell, and at the end of the run, say 72 hours, you can take that same sample off that flow cell and put it onto a second flow cell, run it for another 72 hours, and then take it off that flow cell and run it to a third flow cell. By that time, you will have depleted the amount of your library, but this is the same library being run on 3 different flow cells. So you can actually suck your sample back off a used flow cell and load it onto a new flow cell. It's called a library recovery protocol. So RNA sequencing, Warren was spot on earlier that we're in that nice sweet spot now that the cost is there, especially on a Promethean flow cell where you're going to generate anywhere from 150 to 180 million reads using the cDNA PCR kit. With the direct RNA kit, we used to generate about anywhere from 7 to 10 million reads with the older kit, but with the new kit that's being released in July and August is early access this year, along with the PCS 114 and the PCB 114 upgrade. Those should be released between July and September. I don't have an exact date in those. Those are being developed, but the direct RNA will be released in July, August. This has a newly modified motor protein, because it's moving in the opposite direction of the DNA motor. So it has to be an RNA specific, as well as the pore. The pore has been tightened up as well to have a narrow pore through the middle. So we get better resolution of the RNA. So the accuracy goes from that 94 to 95% raw read accuracy. Now we're up around 97% raw read accuracy, which is going to help a lot. And also help nail down the RNA modifications. And the PCB 114 gives you 24 barcodes, which is really nice if you're going to be doing the RNA-seq experiments to get full-length isoforms. And this is just showing you where we've gone from that about 94, 90, even 93 for the RNA 222. So we're at 96 accuracy for the RNA 004. This is going to be a fantastic kit. So you're going to be getting about 30 million reads on a Promethean flow cell. And again, we have a decision tree of how much input do you have, how much output do you want, do you need PCR, no PCR, et cetera. And I said for the direct cDNA, we do have a protocol that is on our website that uses a modified version of just the standard cDNA kit. You just do an RT step and then prepare it using the ligation chemistry with the kit 114. So DNA shearing. Why is this important? Because you want to be able to fragment to get output or fragment to get the bioinformatics that you want at the end or the analysis that you want. So there's multiple ways of shearing. G-tube is one of them, but G-tubes tend to be rough and shear a smaller amount of the sample or smaller fragment size. So you can see if you go to like even 2,500, you're shearing around 10 or 12 kb. The megaruptor shown here on the right gives you much more control over fragment length. It just has a lower throughput.
So yeah, that was actually March, but I'm a bit slack. So anyway, it's good.
Now,
On
just a
15,
second.
when are you? Oh, the whole
Keep it. Sam, could you, Sam,
I am a part. Yes, I'm more interested anyway.
He shares.
sorry about that. I'm sharing an office and someone just took a call. Okay, so with sharing, a couple of things will affect the library prep or the run of the flow cell. So if you're sharing like here on the left, we're trying to share around 12 kb. Ordinarily, you'd get around 130 gigs on a Promethean flow cell. But if you get this high molecular weight tail, this can reduce your output. And likewise over here on the right, where you get a very high molecular weight tail on a 37 kb sharing N50, this will also reduce your output. And this long tail, even though you get these nice long reads, you can see up here up to 300 kb, this will reduce your output. What you really want to look for in your sharing profiles is something like this, where we got this nice sharing from a megaruptor, where we have no low molecular weight tails, and we don't have that long of a tail out front. So something like this will give you the maximum output. And I'll show you some run reports on this. And again, here, if you can see, sorry, someone's trying to ring me on Slack.
Also, Mike, can you just speak up just a tiny bit too?
Yeah, I just had too many people trying to ring me at one time. That's a choice that I should have traveled there in person. I wouldn't be interrupted. I'd rather be there in person, trust me. But I look forward to being there in person to support Claire and Justin get that stuff up and running. So it sounds like I'll be scheduling a trip over there soon. So Claire, this is important for your human stuff. And you can see over here on the right where you get this a little bit of a tail, but not too bad of a tail. And this runs really, really well. And I'll show you what these run reports look like. And this is just showing you that different inputs in that sharing size and how it affects output. But let me just show you the case studies, which is more important. This is a human sample, and this was sheared supposedly to 34 kb and 50. But if you look at the run profile over here on the left, you can see this really high amount of low fragment size, and then the tail goes all the way up to 100 kb. Now, this was loaded probably 7 or 10 femtomoles. Our library prep says to load the LSK114 on human samples or mammalian samples at 10 to 20 femtomoles. And I have found through empirical working with my high throughput groups in human samples that this is way too much. So what I get people to do is, you can see here, you should be getting 100 GB out of this, no problem, but we've only generated 70 GB of data. The N50 is fantastic, but with better shearing, we can take the same sample, improve the shearing, and now you can see how we've got this nice drop on the left-hand side of this fragment size. We've gone from 71 to 116 GB, which is guaranteed to have more than 90 GB of past reads. This is the total estimated reads. And you can see here we got much better, not quite as fast as a drop off. So if we go back to the previous slide, you got more rapid drop off because the blocking of overloading, as well as the super high molecular weight. We've reduced the molecular weight only a little bit, but the right-hand leading edge of this fragment is much better. So now we've even bumped our N50 up from 34 to 38, and we still got our 30x coverage is what we're trying to aim for most customers. And if you want to have a system where you just walk away, you do that 10 kb sharing, and just with only a single load at 7 femtomoles or 10 femtomoles, we now can walk away and get 140 gig of estimated reads. And if you watch this flow cell at around the 61 hours marks, you can probably get another 20 or 30 gigs because it can run for 96 hours now with these R10.4.1s. I've showed this slide for the main reason is that if you have a very difficult sample.
Can I get you to speak up as much as you can? Because the speakers are not.
Is this better? I just hold the mic right. I just hold the mic right on my mouth. It's just right at my chin a minute ago. The other one to point out is that if you've got a blocky sample, and I'm sure Annabelle and Nikki will share this with you, this is a macadamia tree. And as one customer joked, this was one tough nut to crack, is that just the carbohydrates and the polysaccharides in the DNA will block the pores. The flow cell is running perfectly fine over here on the right, looking at the pore scans. So it's just something that you need to work with Nikki, myself, Annabelle, and also go to the community and look at the extraction and see what people have done to get rid of these polysaccharides. There's plenty of stuff on the community website to help you. Under Nanopore Learning, there's a lot of videos, so go to the lesson libraries and you can see plenty of stuff to watch. There's a metagenomics course. There's data analysis course. The main thing you're going to find for our protocols is under the documentation tab, and under there you can see you've got stuff about sample storage, extraction protocols, DNA RNA handling, library preps, and there's also documents in there about the number of PCR cycles, as you can see here. So there's quite a lot of rich information here to help you be successful. There's also the Nanopore know-how. As I mentioned earlier, the extraction is critical for nanopore sequencing. So we've got 25 sample types, 70 some odd protocols. So just use our resources that are there for you. So, and then under support, if you have any issues, you can always write into support, but there's also some frequently asked questions there as well. Now, in summary, Nanopore has been fantastic for whole genome sequencing, as you saw today. It's great for de novo assembly. It's great for structural variation. You can even phase that. You can also phase the methylome. It's great for epigenetic methylation. It's great for scaffolding and finishing genomes. You no longer have to do hybrid short and long read. If you've got long read, if you've already got short read, you can always just add the long read to finish it off. The targeted sequencing, we've got multiple approaches. We've got amplicon. We've got a hybrid capture approach, which you can do with any platform, but it works really, really well with Nanopore. We've got a CRISPR-Cas9 approach, which we're about ready to release the new version. And there's going to be a single plex, which is the standard protocol, but based on the Kit 14 chemistry. And there's now going to be a 24 plex being released later this year on the Kit 14 using exonuclease digest at the library. So you can multiplex up to 24 samples. And of course, the team at Auckland is really great at the adaptive sampling. And then transcriptomes, I just want to summarize that I think this is the best in class these days because every read is the isoform, as Warren pointed out earlier in his talk. And then metagenomic, of course, just gives you the full assembly of your many of the pathogens or the bacteria that are in your sample. So to me, it's the best platform for doing metagenomics as well. So all that in summary, Warren's already showed this highlight table that just we're basically the most complete platform for sequencing. We look forward to supporting you in your work and your cool biology. And on that note, I just want to make a plug that our Nanopore community meeting in the US will move from New York to Houston on the 6th of December, but we're now going to have a regional Nanopore community meeting in Singapore on the 27th of September, if you're interested. With that, I say thank you. I will stop sharing and answer Warren and I can answer any questions. And next time I'll be there in person.
I was just glad it wasn't someone else on Zoom and it was actually in your office.
No, I share an office on Mondays and Wednesdays.
Any questions for Mike? Okay, and any on Zoom? Thanks, Mike. That was a lot of good overview of like all the different kits and capabilities and kind of hopefully everyone found something that they were like, oh yeah, that's what I want to do.
Yeah, I think the main thing there is the handling of the DNA. It's got to be done right, because if you don't really prepare your library properly, as I showed you by those examples, it will reduce your output and reduce your N50s, and you'll be disappointed.
We actually I had some slides up from Annabelle, which I removed at the last minute, but we've definitely seen that here. So the size can really impact your yield, both good and bad. So
And overloading. So I did a titration when I did some of my own training in Oxford, where we took the kit 14 in the early phase before it was released to you guys, I mean, to the public, is that we did 7 femtomoles, 15 femtomoles, and then 25 femtomoles of, this is just genome in a bottle DNA from the cell line. And we saw a decrease of N50 and a decrease of output accordingly. 7.5 femtomoles was like the perfect output. And then as soon as we went to 15, we lost about 10 to 15 gigs of output. And then again, another 5 or 10 gigs of output where we went to 25 femtomoles.
Yeah. So that's why it's good to get in contact with your core facility who do these
Ex-
audits all the time, and we can help you with that. Okay, well, with that, I know we kind of are over time, and I think there's another group coming in shortly. So I want to just really thank everybody for joining, especially on Zoom. I know we've got a large group on Zoom, and everyone who came today. And we'll just maybe take some questions after. We're going to have lunch downstairs if anyone wants to join. And I really appreciate everyone coming. So thank you all. If there's anything on the chat.
Yeah, I'll check the chat as well.
You're welcome to stick around on the chat and ask questions too if you want. And take snacks for later, because there's a lot of leftovers. And thank you to our speakers.
But DNA, I don't know. No, I have not been yeah. Yeah, so we're looking at my experience. I got pretty much 90 of that. There was not even something. Yeah, that the short reads that it's just I'll be it's really great putting up that and it's see honest yeah it's not even really interesting.
I can show you a slide basically.
So as you said, you can either but if you share it, maybe it's fine.
it's been really good. And I mean, it's not to say, but yeah, you don't have to outside solution, that's still getting like the great amount of data. It's just that you might need to be able to use that for close up. Yeah. Again, besides the actual.
That's what we're ready. Yeah, things.
Yeah, I think that's what we said, because we had a lot of work to say, but sure is included, and yeah, it's on the side. Yeah. Sorry. Thank you. Yeah. A tiny.
Yes.